The availability of trace elements, particularly iron, plays a key role in regulating the abundance of phytoplankton. Iron, as a limited nutrient, is important for nutrition in microbes as it is needed for many processes, such as the synthesis of chlorophyll and the transport of electron proteins. Phytoplankton is the base of the marine food chain, and as such it supports a wide variety of marine life, from microscopic organisms to marine mammals. Through the process of photosynthesis, phytoplankton helps remove carbon dioxide from the atmosphere. There is now a growing body of evidence to suggest that marine mammals could play a role in boosting primary productivity in the surface layers of the ocean. In the past 200 years, whaling has caused a decrease in the populations of large cetaceans. The abundance of many species was drastically reduced in the 19th and 20th centuries. Although certain populations are recovering, it is estimated that populations pre-whaling could be around 10 times higher than modern populations. As whales are suspected to play an important part in the carbon cycle through enhancing primary production, their reduction in abundance could affect the carbon cycle. In 1983, Canwisher and Ridgway claimed that marine mammals played a key role in lifting nutrients from deep waters to the surface layers of the ocean through release of fecal material that disperses rather than sinks. This was the first time that this claim cropped up in scientific literature. In the paper Polar Ocean Ecosystems in a Changing World, it was proposed that a decrease in top predators, such as whales, could have cascading effects on other levels of the food chain. It was suggested that a decrease in whale populations could increase the populations of other species that prey on krill, such as seals and penguins. This paper highlighted that the assumption that ocean ecosystems are entirely controlled by bottom-up processes is flawed. It demonstrated this by pointing out that fluctuations in the populations of key predators seem to influence the populations of prey. However, the paper concluded that it was difficult to find a trend between the population of krill and other predators. As such, a key question that we must ask is, are declining Antarctic krill stocks a result of global warming or the decimation of whale populations? In Smetasek 2008, the paper presents evidence from previous studies that shows a decline in whale populations coincided with a decline in krill populations. Evidence presented shows that krill populations had declined by about 80% over the past 30 years. This is contrary to what would be expected to happen, which would be a trophic cascade in the form of an increase in krill populations. It is considered that the decrease in the population of whales could increase pressure from smaller predators such as penguins and seals, as more krill would be available to them in the absence of a large predator. This is feasible, as Smetasek states that a previous paper by Laws et al. Estimated that whales had an annual food demand of 190 million tons of krill. However, this explanation is insufficient, as seals and penguins constitute less than a tenth of the biomass of baleen whales, and as such are unlikely to have been serious food competitors. Smetasek concluded that as the biomass of one blue whale was equivalent to that of 30,000 Adelie penguins, there would be too great of a discrepancy between the demand of great whales and that of all other krill predators combined for them to be responsible for the decline of krill. This could also be applied to fisheries. Smetasek also concluded that the retreat of glaciers is unlikely to be the main cause, as krill live in regions of low ice cover throughout the world. Smetasek argues that the food chain, diatom to krill to whales, was historically maintained by whales. Smetasek suggests that the decline of whale populations is the primary causing factor in the subsequent decline in krill populations. It is theorized that through the consumption of krill, whales consume large amounts of iron and other limiting nutrients and then release them into the surface layers, promoting phytoplankton blooms. As such, in this theory, whales recycle limited nutrients, particularly iron, back into the surface layer to be used as an energy source for the phytoplankton. As such, this increased prey source would promote the abundance of krill. Krill, then, consume iron-rich diatoms, which in this paper theorized to be stored in their tissues, which is transferred into the surface layer through the whale's consumption and excretion. He presents evidence in the form of satellite images, where productivity in areas that were previously occupied by whales and krill has significantly decreased. The spread of sulps into areas previously occupied by krill is also presented as evidence for a decline in productivity, as sulps are said to prefer areas of low productivity. In the study, Southern Ocean Iron Fertilization by baleen whales and Antarctic krill, fecal samples from whales were collected from several different locations using trawling nets deployed by small boats. Overall, 27 samples were collected from 4 species of baleen whale. Examination found that the fecal iron content was approximately 10 million times that of Antarctic seawater. This supported the hypothesis by Smetasek that whale feces contained high amounts of iron. This study was the first to demonstrate that whale fecal matter contained high amounts of iron, and its findings suggest that the fecal material could be used as a fertilizer. The iron content of 7 species of krill was also examined, and krill tissue was found to have high levels of iron content. Based on this, Nicole et al. Calculated that the Antarctic krill population contains 24% of the total iron in the surface waters in its biological range. In the study, iron defecation by sperm whales stimulates carbon export in the Southern Ocean. It was suggested that through defecating iron-rich liquid feces at the surface area, whales stimulate primary production, thus exporting carbon to the deep ocean. This study estimated that Southern Ocean sperm whales defecated 50 tons of iron into the surface layers of the ocean each year. It was demonstrated in this study that Southern Ocean sperm whales stimulate the export of 4 times 10 to the power of 5 tons of carbon per year to the deep ocean, and respire only 2 times 10 to the power of 5 tons of carbon per year. By enhancing new primary production, the populations of 12,000 sperm whales in the Southern Ocean act as a carbon sink, removing 2 times 10 to the power of 5 tons more carbon from the atmosphere than they add during respiration. It was estimated in this study that the hunting of sperm whales in the Southern Ocean decreased iron inputs into the photic zone by 450 tons each year. Lavery et al. Concluded that whales increase primary production in the photic zone and play an important role in the redistribution of nutrients through their fecal matter. They suggested that whales could have a strong influence on the global carbon cycle through phytoplankton growth, as phytoplankton play an important role in storing atmospheric carbon sequestration. Lavery suggests that whales may be helping with iron fertilization in iron-limited oceans. In another study by Lavery, it was found that blue whales overall contribute a positive amount of net carbon. This study further concluded that whales fertilize their own feeding grounds, sustaining the growth of their prey, krill. However, evidence to conclude this was from the previous study. The whale pump. Marine mammals enhance primary production in a coastal basin. McCarthy et al. Came up with this model for the whale pump, which illustrates how whales transfer iron through consuming iron-rich prey in deeper waters, and then migrate back to the surface and defecate it into the surface layer, boosting primary production. While the transfer of nutrients in the ocean is usually downwards due to marine snow, in this way, whales transfer nutrients upwards. Therefore, whales are often referred to as ecosystem engineers. They undertook this study in the Gulf of Maine, which largely consisted of the input of nitrogen, another nutrient that limits primary production, into the water by whales and seals. They found that the input of nitrogen from whales and seals was greater than the input from all the rivers combined, replenishing 2.3 times 10 to the power of 4 metric tons of nitrogen annually to the Gulf of Maine's surface layer. From this study, they concluded that the frequent release of faecal plumes brings essential nutrients to the photic zone, and that, according to their data, marine mammals can enhance primary production. Based on all the data in this study, from the estimates of the abundance of whales pre-whaling, estimates were made to see the levels of nitrogen from different sources into the Gulf of Maine. The results show that pre-whaling input from the whales was very significant, exceeding other points of input. The biochemical role of baleen whales and krill in Southern Ocean nutrient cycling. This study analyzed iron and other trace elements in baleen whale feces and krill muscle tissue. It was found that, in krill tissue, metal concentrations were between 20,000 and 4.8 million times higher than the seawater concentration. In whale feces, metal concentrations were found to be between 276,000 and 10 million times higher. Preliminary investigation into the stimulation of phytoplankton, photophysiology, and growth by whale feces. A study by Smith et al. In 2013 found that the feces of pygmy blue whales stimulate photosynthesis and growth in 3 phytoplankton species: Dunaliella tertiolecta, Chatoceras pendulus, and Phycistis antarctica. In this study, increased biomass of the 3 species correlated with increasing concentration of whale feces. However, a limit in this study is that the feces used were collected from the coast of Western Australia, and as whales in different regions have different dietary compositions, it would need to be undertaken to see if results are consistent in other areas. The results of this study are in line with the conclusions of the aforementioned Roman and McCarthy in 2010. The original paper by Canwisher and Ridgway states that the fecal matter of marine mammals could increase primary productivity in the surface layer. However, the paper makes no attempt to explain this claim, presenting no evidence nor linking to any other papers that support the claim. This was the first time that this claim cropped up in scientific literature, and did not start to find any significant support until Smetasek's paper in 2008. Smetasek's theory suggests that whales would have played a heavy role in historically promoting the abundance of krill through recycling waster products. He suggests that in conditions before industrial whaling, the mechanism would have been more significant in the marine environment, and that whales would have released far larger amounts of iron into the photic zone. However, while this claim is based on sound reasoning, the lack of historical records on this subject mean that it is not supported by quantitative evidence. A limit in testing this hypothesis is that human activity has permanently changed the dynamics of the community, meaning that it is impossible to conduct further research to support certain parts of Smetasek's claims. A drawback in this study is that the iron inputs from thermal vents are not accounted for. It is possible that a decrease in input of iron from this source might have contributed to a decrease in productivity. As such, I propose that there is a possible link between the decrease in primary production and whaling that could be found in the decreased abundance of whale fools falling to the sea floor. Before industrial whaling, whale fools at the bottom of the sea were substantially more abundant. To be precise, a study calculated that prior to whaling, whale fools were 6 times more abundant than at present. Whale fools have long been considered as stepping stones for vent and seep taxa. However, it should be noted that the current research does not confirm that whale fools necessarily play a role as a stepping stone habitat between hydrothermal vents. It is likely that whaling influenced the biodiversity of whale fool communities, and may have led to the first anthropogenic extinctions in the deep sea in the form of whale fool specialists. It may be possible that the reduction of whale fools as stepping stone habitats between thermal vents had a knock-on effect on inputs from thermal vents. We will touch more upon this subject in the second part of the video. Back to the subject at hand. Supplementary evidence is presented in eyewitness reports from a survey on krill distribution in the Southern Ocean by the HMS Discovery in the 1920s and 1930s, where it was noted that the region with the highest densities of krill coincided with that of the main predator, baleen whales. However, as none of this was quantified or recorded, the validity of this evidence is very limited. Another drawback that Smetasek admits is that his hypothesis could not be undertaken on small-scale experiments, such as enclosure experiments or field observations. This means that any future studies attempting to prove his theory would have to take place over a larger scale. For further context on the studies Smetasek used to make calculations, it has been estimated by previous studies that whales consumed 190 million tons of krill per year. Smetasek found that through krill consumption, whales convert around 7,600 tons of iron in krill into iron-rich feces every year, around 12% of our estimate of the current iron content of the surface layer. Based on Smetasek's reasoning, larger populations of whales pre-whaling would have led to an enhanced primary production, and this larger food source would have potentially supported bigger krill populations. Iron defecation by sperm whales stimulates carbon export in the Southern Ocean. The results contribute to the idea that whales concentrate and retain trace elements vital for primary production in the surface layer through consuming krill and then making iron bioavailable to phytoplankton through excretion. The results show that krill can contain large amounts of trace elements, acting as a reservoir, with whales acting as a mechanism of dispersing these trace elements in the photic zone. However, the study highlights that no study to date has been able to determine the bioavailability of whale feces. For additional context, at first in the Southern Ocean, limited growth was thought to be the result of limited light availability. However, it was found by Boyd et al. That iron limitation limits phytoplankton growth, and iron input from upwelling in the deep water was found to be weak. It is believed that whales present nutrients needed for photosynthesis by phytoplankton in high nutrient, low chlorophyll waters, like the Southern Ocean. In the Southern Ocean, baleen whales act as a major predator of Antarctic krill. Through daily vertical migrations, krill transport limiting nutrients and stimulate primary production. During the short summer season in the Southern Ocean, adult blue whales consume approximately 2 tons of krill per day. During the summer, whales consume a larger amount of lipids, meaning that most of the iron is excreted out. Characteristics of whale feces. Most aquatic organisms have fecal matter that occurs in the form of discrete pellets, which either immediately sinks or ends up being carried away by currents, usually being spherical or cylindrical, or formed into strings, depending on the organism. These physical characteristics result in the sample rapidly sinking or dissipating into the water column. The characteristics of whale feces are different. Cetacean feces have been described as a slick stream of particulate matter, or a plume. These characteristics result in the plume quickly dispersing and staying present in the surface layer, rather than immediately sinking. In summary, as whales slow down non-essential biological function when diving, whale feces are excreted in a quasi-liquid state. Due to this, the defecation stays present in the surface layers of the water. The bacteria present in the defecation causes an increased residence time of the nutrients due to their high uptake potential, thus allowing for blooms of phytoplankton. Conclusions. There is strong evidence to suggest that whale feces contain high amounts of essential nutrients, such as iron, that can boost surface production. However, lack of data on the bioavailability of these trace elements released by whales in excretion makes it difficult to say what percentage of it ends up being used in primary production. While studies such as Smith et al. Do demonstrate that whale feces can stimulate growth of phytoplankton, there is a lack of research that demonstrated this on a large scale. However, the fact that whale feces do transport limiting nutrients in the photic zone is well supported, but it is hard to quantify how this may have been affected by commercial whaling, as research was not carried out in pre-whaling conditions. Therefore, it is hard to calculate the exact loss to primary production from whaling. The loss of marine mammals can potentially affect rates of primary productivity. Historically, whales would have been the main consumers of krill, and as such would have had a greater contribution to the recycling of nutrients in the ocean. Commercial whaling has affected the cycling of essential nutrients in marine ecosystems. While editing this video, I came upon two recent studies, which I will very briefly summarize. The first study, by Pearson et al., done in 2023, quantifies how much carbon is removed by whales, taking into account whale falls, the aforementioned whale pump, the great whale conveyor belt, which is the process of whales transporting vital nutrients through long-distance migrations, which often exceed thousands of miles traveled. This diagram on screen, which is also from the study, illustrates how whale migration stimulates transport of nutrients between nutrient-rich feeding grounds to nutrient-poor breeding grounds. For species such as humpback whales, right whales, and grey whales, breeding grounds are often found in shallow areas with low nutrient concentrations. This means the nutrient subsidies that whales bring into these habitats support fish, scavengers, and also any benthic macroinvertebrates. The study also contains this table, which shows the amount of carbon fixed, stored, exported, and sequestered by these activities in pre-industrial whaling and modern-day whale populations by different whale species. The study concludes that whales are capable of carbon dioxide removal, and restoring whale populations to previous levels is a potential carbon removal strategy that could be used for climate change mitigation. The second study I found while editing was by Monreal et al., which was actually published this year in January. This study explores how iron and copper recycling was reduced by industrial whaling. This study states that the transport of micronutrients by whale fecal plumes was reduced by 90% as a result of industrial whaling. This is a very sophisticated study, and so I suggest you go and read it yourself after the video, especially if you want to understand things from a more chemistry-based perspective. I want to thank you for watching this video. Hours has gone into finding studies, script writing, voiceovers, editing, and preparing the video for upload. As such, I would greatly appreciate it if you could like the video, share it with your friends and colleagues, and also subscribe for future uploads.